Right now, wherever you are, I just want you to take a look at your own hands. Seriously, just look at your skin for a second. It looks totally static, right? Well, if we could somehow do a hyper-realistic microscopic 3D zoom right past your fingerprints, deep into the tissue, you absolutely wouldn't see something static. You'd actually witness this chaotic, graphic landscape. We're talking flaking dead skin cells shedding away, pulsating capillaries pumping resources, and just this relentless, invisible hum of tissue rapidly rebuilding itself. So welcome to today's Explainer. We're going to unpack the visceral, hyperactive biological events happening in your body this very second, basically the ultimate copy and paste mechanism.
37 trillion. Now that is a massive number. Every single human being starts out as just a single, solitary cell. Literally just one. But today, you are a walking metropolis of roughly 37 trillion of them. So how do we get here? Well, cell division. It's the mechanism that built you from scratch, but it's also this aggressive machinery that replaces worn-out tissue and heals your everyday injuries. Just think about the last time you got a paper cut. I mean, on a macro level, it's just a minor annoyance, right? But on a microscopic level, that is a catastrophic canyon of torn flesh. To fix it, your body actually has to rapidly clone millions of somatic cells, physically stacking them up to build a fleshy bridge across that microscopic gap.
Okay, let's move into Section 1. Two main pathways, mitosis versus meiosis.
So human biology utilizes this remarkably strict, organized system to split cells, and it operates on two entirely different pathways depending on what the end goal is. First, you have meiosis, which is a highly specialized operation reserved exclusively for reproduction. It creates those entirely unique gametes, sperm and egg cells that contain only half the original genetic material. But then you have mitosis, and that's the everyday workhorse we're really focusing on today. Mitosis is honestly like an aggressive, relentless industrial cloning factory. Its entire job is to create an exact duplicate, a perfect one-to-one biological clone for your somatic cells, actively duplicating tissue to keep you functioning and intact.
Section 2. Preparing to divide. Interphase. You see, a cell doesn't just divide constantly. It actually spends most of its life right here in interphase, just doing its normal job while quietly, meticulously duplicating its entire genetic code in the background, basically stockpiling resources for the violent split ahead.
Once that DNA is perfectly copied, the cell steps out of interphase and into PMAT. Now, PMAT is just a simple acronym representing the four main operations of mitosis. And let me tell you, this is not a gentle process. It's an aggressive, highly coordinated mechanical sequence of biological teardown and reconstruction that transforms one single cell into two. We've got prophase, metaphase, anaphase, and finally, telophase. Let's really dive into the visceral 3D reality of how your body physically tears a cell right down the middle. We start things off with prophase. Inside the nucleus, you have this chaotic, tangled soup of chromatin, and it aggressively hypercondenses. It winds up incredibly tightly into these very neat, distinct, thick, X-shaped structures called chromosomes. It's a visually striking transformation, for sure. And at the exact same time, the cell deliberately tears down its own defensive walls. The nuclear envelope, which normally quarantines the DNA, completely dissolves, basically releasing these thick chromosomes out into the open fluid of the cell.
Next up is metaphase. In this phase, these heavy-duty microtubule cables, which are known as spindle fibers, literally shoot out from the extreme edges of the cell. They physically harpoon those thick chromosomes, latching right onto their centers. Then, with relentless tension, these microscopic cables force the chromosomes into a perfectly symmetrical line, dragging them right down the biological equator of the cell.
Then we hit anaphase, and this is where the dynamic mechanical action really kicks into overdrive. Molecular motor proteins crank into gear and literally rip those X-shaped chromosomes in half right at their center. The microtubule cables forcefully retract, dragging these raw, identical genetic clones through the cellular fluid, pulling them all the way to the complete opposite poles of the cell. It's pretty wild to think about.
Following that is telophase, which is basically the reconstruction phase. Now that the identical DNA halves are safely segregated at opposite poles, the cell itself begins to warp and literally cave in on itself. New nuclear vaults are rapidly constructed, throwing up these protective walls to quarantine and safeguard the newly separated DNA clusters. So, at this point, the cell has successfully built two brand new, identical command centers.
And that brings us to Section 3, the final split, cytokinesis. Cytokinesis is the actual physical severance, the grand finale really.
Right at the cell's equator, this microscopic drawstring called a contractile ring forms. This ring of proteins ratchets tighter and tighter, literally choking the cell membrane inward, forming a deep cleavage furrow. It just continues to squeeze with immense microscopic force until the membrane fuses and entirely snaps into two. So what was once a single, solitary cell is now two distinct, independent, perfectly healthy genetic clones.
This relentless industrial machinery is the exact reason you survive everyday wear and tear. I mean, think about it. When you get a scrape, that persistent itch of a healing scab, that is quite literally the physical sensation of millions of these cycles firing off. It's millions of your cells aggressively hypercondensing DNA, harpooning chromosomes, and choking themselves in half just to knit your torn flesh back together.
As we wrap up this explainer, I want you to look at your hands one more time. Really think about the last few minutes we've spent together. How many millions of your very own cells just forcefully divided, cycled through PMAT, and beautifully replaced themselves while you were just sitting here watching? The universe inside you literally never stops. Keep that curiosity alive, and I'll catch you in the next explainer.